And it's all very well writing out a genome, but how the hell do you know what it means? You've just got this string of letters, A, C, D, G, A, C, D, C, 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 T, A, G, G, A, C. What does it mean? I'd been thinking about embryology for a very long time, even before I had children. I taught embryology at medical school in the University of Bristol, and I'd done a little bit of embryological research as an undergraduate as well. It was a subject that utterly fascinated me. And it is an incredible story. I think sometimes with these science stories, we know things so well, and we've known them for such a long time, and we can't even remember when we learnt things, that we somehow lose the wonder that's attached to them. Each one of us started off as a single cell. Just one cell. You wouldn't have been able to see it on the tip of your finger unless you knew it was there. It was quite a big cell. It was your mother's egg, your mother's ovum. So you've turned from a single cell into you, into this incredibly complex organism with lots of different tissue types and cell types, and a beating heart and a brain that thinks and contains emotions and memories and knowledge. How did all that happen? How have you gone from a single cell to being this incredibly complex organism? I got really frustrated looking at pregnancy books that would kind of go, and they'd have a bit of detail about conception, and then they'd move on, and there'd be a ball of cells, and then suddenly there'd be a picture of a baby. And you go, but something interesting happened in between. How did you get from this sort of ball of cells to a baby? Well, actually, going back to conception, that in itself was a mystery for a very long time. Again, it's a sort of thing we all know, don't we? I think everybody in the audience here knows how conception happens. I do always have to check, though. I was giving a talk in Ireland about a month ago, and there were some very little ones in the audience. So I had to use an astonishing amount of innuendo to get my points across. And I think the adults in the audience must have thought I was an extreme prude, because there were certain words I wouldn't mention in front of these tiny little ones, and certain concepts I wasn't sure their parents were keen for them to learn at such an early stage. Anyway, we know how conception happens. It was a mystery for millennia, for centuries, for millennia. Aristotle in the 4th century BCE tried to work out how this could possibly happen. And he wrote a book called On the Generation of Animals. So he was interested in where humans and other animals came from. And he obviously could ask people about it. I mean, it's interesting, isn't it? Think yourself back to the 4th century BC. What information is possibly available to you? What information is not available? Anything you need a microscope to see, you're not going to know about. So you don't even know about cells. You can obtain information by talking to people. So I think Aristotle talked to a lot of people, and he came to the conclusion that nobody'd had a baby without having sex. So sex must be something to do with it. And you go, he's meant to be a genius. Does it take a genius to work this out? But actually, hang on a minute. It's not that obvious. When a woman conceives, she doesn't look immediately pregnant. And actually, the product of the conception doesn't pop out until 9 months later. So it's a pretty clever thing to work out, that there's a connection between sex and babies. And not only that, we are, I think I'm right in saying, the only species on the planet that knows that connection. That's quite interesting, isn't it? We are the only species that knows that sex and babies are somehow related. All the other species just do it for fun. So having made this momentous discovery that sex and babies were connected, Aristotle then went on, that wasn't enough for him. He's like, right, okay, so there must be something. What materials come together to make the baby? What materials create this new life? And he said, well, I think it's got something to do with semen on the part of the male. Again, not really a genius stroke there. And then he was a bit lost when it came to the woman. And he said, well, maybe... Sorry, I need to fix this. A bit of adjustment going on here. Maybe there's, on the female side, something there too, and the thing that is likely to be is menstrual blood. Because when a woman becomes pregnant, she stops menstruating. So he had this very peculiar idea that, again, to us, sounds very, very odd, and we laugh at it. He called it epigenesis, and it was this idea that you took semen, which to him was a homogeneous milky fluid, you mixed it with blood, which was a red fluid, homogeneous fluid, and out of that, you got a baby. He was satisfied with that. So, yes, I've nailed it. Good. Epigenesis. And this was the enduring theory of conception for centuries. Centuries and centuries and centuries would go by, and we'd go, yeah, that's how it happens. Mix a bit of menstrual blood together with a bit of semen and baby. It kind of fitted into the philosophy at the time, this idea of spontaneous generation. We peer back into the past and try to understand when life on Earth first emerged, and we're looking most intently now at those deep sea vents at the bottom of the ocean, which are almost experiments in bringing together different molecules, and we think that life may have started there. But to the ancient Greeks, all the way through to the Middle Ages, people believed that life just sprang into being all the time, that spontaneous generation, that animate matter could emerge out of inanimate matter. That was just an everyday occurrence. Of course it was. Everybody knew that living flies could come out of cowpats. Everybody knew that living flies could come out of a dead piece of meat. Nobody'd seen a fly laying eggs. So there was this idea that flies just sprang into being. So Aristotle's idea in that context doesn't seem so strange, that inanimate fluids could somehow magically produce something living. No amount of sitting around was going to solve this. And this is interesting when we look at the history of science, that we have people theorizing, but actually we need evidence. And the evidence that was necessary to get closer to the solution of this question of how babies were made involved being able to see what was there. And so it involved being able to extend the optical abilities of the human eye. In the 16th century, we start to get lenses being crafted and magnifying glasses being made, and people start to stack these lenses. Galileo stacked them in order to bring objects that were far away much closer, so that he could see the details of planets. But he also experimented with bringing tiny things much closer as well, using the same technology. So we don't know if he was the inventor of the microscope, but it was certainly happening around that time. And this is what early microscopes looked like. This is an occhiolino. This is the Italian word for microscope. Much nicer, I think. In Britain, microscopes were used to provide the material for the first ever publication of the Royal Society in London. And this is Robert Hooke's Micrographia, which is a fantastic book. It is a coffee table book and then some. It is about that big. And we've got a copy of it in Birmingham, where I work now, in the University of Birmingham, in our fantastic research collection. And I spent a lovely afternoon leafing through it. I mean, look at that flea, it's fantastic. So Robert Hooke was putting all manner of things underneath his microscope, fleas and bees and nettle leaves, and looking at them in more detail than anyone had ever seen before, and then drawing them in this book. And people were astounded to see how much detail there was in biology that nobody had ever seen before. On the other side of the North Sea, another gentleman, Antony van Leeuwenhoek, was also making microscopes and using them to look at things. His microscopes looked very different. This is one of his microscopes. He was doing something rather interesting. Rather than what Robert Hooke was doing, which was to take things we already knew about, like a flea, and stick it underneath the microscope and see it in more detail, Leeuwenhoek was interested to see if there were things that we hadn't even imagined yet about the world. So he put things underneath his microscope just to see what was there. And so he did things like putting a droplet of seawater underneath his microscope. And he saw that there were what looked like tiny plants floating around in it. But not only that, there were absolutely minute swimming animals in it. So he was the first person to see zooplankton. He called them animalcules. He did look at the human body as well. So he was the first person to see the structure of spleen and liver. He was the first person to see red blood cells. So suddenly that homogeneous red fluid of Aristotle's became something that was full of cells. And we would learn eventually that the way that bodies are formed, both plant bodies and animal bodies, are by these units, these cells. So think yourself back to 1677. And there's a lot of things you don't know about the world, including conception. So you have to forget, again, that you know about conception. And you're in this world where, probably it's something to do with epigenesis and Aristotle's ideas. You take a sample of milky fluid, totally leaving it up to your imaginations how you get that sample of milky fluid, and you put it underneath the microscope and you peer down it, and what you see astounds you. And of course, this should be moving. So what you see is a field full of life, of buzzing tadpole-like things rushing around, swimming, thrashing their tails. So he was the first person ever to see sperm. And he thought it was pretty amazing. Not only were there these animalcules swimming around in seawater, there were what looked like animalcules in the human body. And again, you go, yeah, I know about this. I've known this since I was very small. But how incredible. Biology suddenly got very bizarre. How incredible that there are these weird little swimming things. For the half of the audience that doesn't have these weird little swimming things, which are weird, how odd, we do have other cells which are strangely autonomous. I always think white blood cells are a bit strange. There's this kind of army of cells inside us who are kind of just doing their own thing. We've got no idea what they're doing. They'll respond to signals. If there's a bit of inflammation going on, they'll head in the blood vessels, and then they'll crawl out through the edge of the blood vessel and get to where the inflammation is and go and try and tackle it. And you've got no idea this is happening. So this army that's working on your behalf, full of these little autonomous beings almost. So sperm, he wrote to the Royal Society because it was the preeminent scientific society in Europe at the time. He said, look what I found. The Royal Society went, brilliant. That's really interesting. What else could you look at? Neither of them realised that he'd found half the secret of conception. It was kind of weird, but that was as far as it went at that point in time. And then eventually the penny dropped. Now, finding the egg would take much longer, which is bizarre because the egg is so much bigger than the sperm. Sperm are absolutely tiny. The egg is the biggest cell in the female body. It is 0.014 millimeters in diameter. And as I said before, if you had one on the tip of your finger, I don't know why you'd have a human egg on the tip of your finger, and you had good eyesight, you would just about be able to see it. Most cells in your body you haven't got a chance of seeing. The human egg is big. Renier de Graaf nearly found the mammalian egg in 1672. He was looking at rabbit ovaries. Biologists assumed there was a mammalian egg because of the similarity of the insides of mammals with the insides of birds and reptiles. When they looked at birds and reptiles, it's very clear to see an ovary and an oviduct and a little uterus, which is a holding station for the egg, and then out the egg pops out. When you open up a mammal, you see similar structures. So the biologist said, well, we're good at comparative anatomy. We think this is what's going on in mammals too. We think we've identified something which is an ovary, even though the eggs must be very small. If you look at a hen's ovary is quite extraordinary. It's quite a big structure within the body of the hen. And the biggest egg in the ovary is exactly the same size as the yolk in your fried egg. So when you fry an egg, pop it out, or a boiled egg, doesn't matter what you do. Don't scramble it, because then you ruin it. But crack your egg open, have a look at the yolk. That is the egg. That is the original hen's egg. And there was a little bit of DNA on one side of it that then teamed up with the cockerel's sperm if it's going to be a chick, not if it's going to be a breakfast egg. We hope that that's unfertilized. But that whole thing is the hen's egg. So the mammal egg is a lot smaller than that. It doesn't need nutrients to support it inside an egg because you end up with a placenta. So Renier de Graaf nearly found them. He found these follicles which bear his name. They're called Graafian follicles. So this is a rabbit ovary in the end of a rabbit oviduct. And then in 1827, so 150 years after the discovery of sperm, we have Carl Ernst von Baer finding the mammalian ovum. Ta-da! Great. Aristotle, what did he know? So we can get rid of epigenesis. We can say, he was definitely wrong. Semen's not a homogeneous milky fluid. It's got these sperm in it. And there are eggs as well that come out of the ovaries. And the two things come together. And that's how you make a baby. The thing is, though, that Aristotle was a genius. And his question that he was trying to solve still remains. We've got a little bit closer to answering it, but actually the fundamental question is still there. Because the fundamental question is, how do you get all of that complexity of a baby out of simplicity? And epigenesis tried to explain that. A baby is still a lot more complex than a sperm and an egg. So how can a sperm and an egg create the complexity of a baby? We've still got that conundrum. In the 17th century and 18th century, some biologists believed they had the answer. And the answer was that the complexity was already there. That as our microscopes got better, what we'd see is that there was a tiny, tiny baby curled up in the head of the sperm. You see its head with its soft spots, just like that. And then it's somehow activated by the egg and it just grows bigger. This school of thought was called preformationism, and the preformationists fell into two camps, the spermists and the ovists. The spermists believed this. The ovists believed that the tiny baby was inside the egg, and the sperm activated that. Now, this is interesting. This is all happening with a kind of biblical framework to this science. So let's contemplate the ovary of Eve. The ovary of Eve contains within it lots and lots of eggs. And some of those eggs will be male and some of them will be female. We don't need to think about the male ones at the moment, we'll think about the female ones. Inside those female eggs are tiny little female babies, and inside those babies there are little ovaries, and inside those ovaries there are eggs. And half of those eggs will be female babies. So inside those eggs there will be female babies with ovaries, and inside those ovaries will be more eggs, and it just goes on and on and on. So it's called emboîtement, this lovely French word, boxing up, like Russian dolls. This idea that Eve had stacked up in her ovaries all of the generations that were ever going to be on Earth right until doomsday. What an extraordinary idea. And then cell theory came along, and you can't get things smaller than cells. Cells are the units which make up the bodies of plants and animals. So this was clearly wrong, and microscopes got better, and nobody could see a tiny baby, either in the sperm or the egg. So then Aristotle's question comes back, floating back from the 4th century BC. How does the complexity come? Where does it come from? Well, we know about conception now. This is an ovary, a long way away. It's a very strange diagram, this, because there's far too much of a distance between the ovary and the end of the oviduct here. Out comes the egg. It passes into the oviduct. If it's a lucky egg, there's sperm coming up in the other direction. The sperm penetrates the cells that surround the egg. They penetrate the gel-like layer, the zona pellucida, around the egg. And the genetic material from the sperm passes inside the egg. And the egg immediately starts to divide. So this fertilized egg then is going to start changing from being one cell into 2 cells. This happens within 24 hours of fertilization, which actually is relatively slow compared with other animals. And at the same time, this fertilized egg is moving. So it's moving on the inside of the oviduct, helped by the waving cilia inside the oviduct, and which are wafting it closer and closer to the uterus, which is where you want it to end up, so that it can develop into a baby. And what's really fascinating to look at is what's going on inside that fertilized egg. So this is tricky, always a bit tricky when we've got bright lights, because you can't see the detail. The yellowness is stretching right out to about here. And what we're looking at here is an egg. And the genetic material inside the egg is shown in blue. Oh, that's lovely, thank you. So now you can see some more of the yellow, can't you? You can see where the edge is, because then it goes black. So the genetic material inside this fertilized egg is in blue, and the scaffolding, the skeleton of the cell, is shown in yellow. This is a really clever technique. So this is where we're at with microscopy now. You put a lot of things under microscopes and they're just completely see-through, and it's very difficult to see details. You can stain them up, so the science of histology has depended on this. So you can put different dyes onto structures, onto tissues, and they will differentially stain those tissues, so you can see more detail. But actually what we can do now is just send in dyes that will dye precisely what we want them to dye. And we do that using antibodies. So basically you can choose anything in a cell that you want to look at. You can get an antibody to stick to that thing, and then you can stick a dye on the antibody. So here we've got an antibody which is actually attaching to the protein that packages up DNA, and we've stuck a blue dye to it. So that shows us where the DNA is. We've also got an antibody which will go and stick to tubulin, which is this protein that forms a scaffold inside the cell. And we've stuck a yellow dye onto that, So when you look at it under the microscope, they fluoresce, and you get these wonderful images. So this is from George von Dassau's lab, and all his images are online, and you can explore them. It's got some really stunning images. What we've got here is a newly fertilized egg. There's a ball of genetic material from the mother and a ball of genetic material from the father. That genetic material has already doubled up. There's actually enough DNA there for 2 cells, but we're going to have to sort it out a bit better than that. And this is where the skeleton of the cell comes into its own. So the skeleton's forming this kind of spindle in the middle, and the chromosomes line themselves up on it. So we've got pairs of chromosomes now, and then the pairs will separate. So each one of these blobs here represents a pair of chromosomes, one from the father, one from the mother, of complementary chromosomes. And they're moving away from each other. They're moving in this direction and this direction. And the cell membrane is starting to pinch in as well. So we've got this dumbbell-shaped cell, and eventually that's going to split in 2, and you end up with 2 cells. What a wonderful thing to see. And these are actually images which are looking at the ribbon worm, Cerebratulus. So this isn't a human fertilized egg. This is a ribbon worm, a very, very, very distant relative of ours. But the way that it happens in the human egg is similar enough to show you these pictures and to say this is what happens. Then it carries on dividing, so that as we move towards the end of the first week, we've got a ball of cells, and then eventually that starts to take on fluid, and we've got a cluster of cells there which become the embryo itself, and then actually this kind of skin around the outside becomes part of the placenta. And that whole thing eats its way into the lining of the uterus like a little parasite. And the uterus goes, yeah, all right, you can do that. It's designed to do that. It's designed to allow this invasion and then to grow with the conceptus as it develops. Now I am going to have to jump a bit to week 4. So we've now jumped, in fact, 3 weeks on. And, you know, if you wanted to stay here all evening, I could fill in the gaps. Tell you, you could read my book. So the book fills in the gaps as well. During this time, what's happened is that many more cells have been created. Cell division is going all the time, going. But differentiation is happening as well. So cells are deciding what they're going to be when they grow up. They start going off in different directions. They form layers. In fact, the whole thing starts off as a disc, and then the disc curls around to make a kind of sausage. And that's the point at which we reach it. So this thing, which was a kind of flat disc of cells, has now curled around. And it's actually already got a gut tube in the middle here. And the gut tube is communicating with this strange sac on the outside. And this sac is a yolk sac. This is really odd, because you didn't come from an egg that was laid by your mother. You were kept inside your mother's body, and you were supported by the placenta. You didn't need a yolk. And in fact, this is an empty yolk sac. So the yolk sac of a hen is full of yolk nutrients. They're yellow. You know that from your eggs. So the yolk sac of a human is completely empty. Well, it's just got fluid in it. It doesn't have any nutrients at all. There is no reason for it being there. And this is one of these evolutionary echoes in embryology. The only reason that's there is because you had ancestors who laid eggs. And it's just a kind of memory. Somehow it's been impossible to scrub it out of the program of development. Maybe scrubbing it out of the program of development derails everything else. So you've just kind of, for a bit of time, you've just got this kind of weird yolk sac thing hanging around that you don't really need and you don't really want, and eventually it just gets squashed and disappears. But it's there for a bit. Week 5, things start to get interesting. You've got the beginnings of arms and legs. Oh, you've got a tail, another hangover from previous existences, from ancestors that had tails. That regresses as well. So we'd see this a lot in embryology, that things form almost as though they're going to go down the route of becoming something ancestral, and then they go, no. And then they start to regress back again. Something interesting, neck of a series of ridges. Strange. You don't have a series of ridges in your neck, but you certainly had a series of ridges and clefts in your neck when you were an embryo. So those must get turned into something else. I mean, maybe they just disappear like the tail and the yolk sac. In fact, they don't. They get recycled into other things. So another fate of things that kind of come along in embryology is that, and are not needed as such, is that either they could disappear completely, or you just go, oh, I've got this stuff now, I'll recycle it into something else I do need. We get an idea of what those bars and ridges are about if we look at a distant relative, and this is a shark egg. So you've probably seen these washed up on beaches. We call them mermaids' purses. They're not mermaids' purses. They're the eggs of sharks. I've already disabused my children of this. My daughter was quite taken with the idea of mermaids' purses. Right? No, definitely not. She still believes in the tooth fairy, though. I have to put an end to that soon. It's getting too expensive, apart from anything else. So this is what this little shark is going to look like when he comes out. So he's got a series of ridges in his neck. Oh, look, he's got a yolk sac as well. He's got a yolk sac full of lovely yellow nutrients. He needs that. He's not in his mum's tummy anymore. And he's got these series of bars in his neck. And probably now you're thinking, OK, I know what these are now. This series of bars and ridges in the neck. If we look at a grown-up dogfish, which is a type of shark, you can probably just see here some of those clefts in its neck. So these are gills. So that's what those ridges and clefts in the neck of the embryo are destined to become. Really? In a human embryo? Why have you got things which look like you're going to have gills? We don't have gills. We don't need gills. Our ancestors left the water 360 million years ago. Why on earth have we got things that look like gills? Well, again, presumably there's a bit of programming here that means that if you try and scrub out this bit of structure that appears at week 5 in the human embryo, you just derail lots of other stuff. And instead, what has happened is this interesting recycling. So in fact, each of those gill arches appears, and it goes as far as to have a little bit of cartilage inside it, as though it's going to be supporting a gill. It goes as far as to have an artery coming into it, as though it were going to supply the capillary bed of a gill. It has muscles which will open and close the gills, and nerves supplying those muscles. And then you start pushing it off in another direction. So you take all of those components in each of those arches, and in fact there are 5 arches which appear in the human embryo, and you just recycle them into stuff you need in the head and neck. Stuff that your fishy water-breathing ancestors didn't need, but stuff that your air-breathing ancestors, once they emerged onto land, absolutely did need. They'd evolved lungs. They needed an airway to connect the lungs to the outside world. And the airway needed to be supported by cartilage to stop it collapsing, just as gills do. So you take the cartilages from some of the lower arches and you model those into a voice box. Before anybody thought of speaking with a voice box, it's there to keep your airway open. How extraordinary. Higher up, arch 2 and 3, so this is a generic vertebrate embryo, arch 2 and arch 3 make part of your hyoid bone, which is a little bone which anchors the floor of your mouth and is what your larynx hangs from, what your voice box hangs from. So again, something that fish don't need, and you don't need it until you've got a larynx. Something else you need to do when you emerge onto land that your fishy ancestors didn't need to do is to be able to detect vibrations in air. So fish detect vibrations in water through their lateral line systems. That's not going to work. Once you've got things like a Canthostega crawling out onto land, can't feel anything. Can't detect any vibrations around me anymore. So you've got to create something new. You've got to evolve something new. And this is all happening through mutations and structures changing and then natural selection, picking out things which work well and weeding out things which don't work well. And what happens is that, again, things which would have become bits of gills in your fishy ancestors get grabbed and pulled inside the head and end up being the ossicles in your ear. So the hammer, the anvil, and the stirrup, or the malleus, the incus, and the stapes, to give them their proper names, those tiny little bones in your middle ear came from the bits of cartilage in what would have been gills if you were a fish. It's just astonishing. So this is the science of evo-devo, evolutionary developmental biology. It involves following embryological development really carefully, step by step, so that we can see, for instance, when the shark embryo and the human embryo start to go off in different directions when you look at particular structures. And it shows us how form has evolved over time. So finally, I want to introduce you to a very distant relative. We've talked about fish, so our relatives with fish go back about 400 million years ago. And this is a very distant cousin. If you want to look for a common ancestor with a fruit fly, you're going back about 800 million years ago. Of course you've got a common ancestor with this. Everything on Earth is related, so eventually you get a common ancestor with everything. We believe that life had a single origin and that there was a universal common ancestor of absolutely everything, no matter if it's an amoeba or a fungus or a plant or a human. And that's because we all use the same information to build our bodies. We all use nucleotides, either DNA or RNA. So it shouldn't perhaps be so surprising, although it's still a bit jarring, isn't it? I think these little flies that buzz around my fruit bowl are distant cousins. This is all starting to seem a bit weird. They're very different from us, aren't they? They have 6 legs, they've got compound eyes, they've got wings. It's very difficult to look at their anatomy and spot any similarity at all between us and fruit flies. You have to go a bit deeper. And here's the man who started it all off. Thomas Hunt Morgan was working in the University of Columbia in the early 20th century, and he was studying heredity. Now, of course, this is way before DNA itself was discovered. And what Thomas Hunt Morgan worked out was that heredity, or the units of heredity, were in the nucleus of the cell, and not only that, but they were it was on the chromosomes. So he worked out that's where it was in the cell, this stuff. Whatever it was that was passed from one generation to another and enabled traits to be passed from parents to offspring, that's where it was contained. So his breakthrough was necessary before you then managed to work out what the molecule was inside of those chromosomes. So he got a Nobel Prize for that in 1933. And he was very interested in looking at differences between fruit flies and studying Mendel's patterns of inheritance. So he was breeding together different types of fruit flies and looking at the patterns of inheritance that he got. Some of them were natural variants, and some of them were very unnatural variants. And this is one of his unnatural variants. So this is a wild type. This is a normal fruit fly head here, and it's got antennae growing out of its head. And this is a very abnormal fruit fly that's been zapped with radiation in order to produce this mutation. This one's got a leg growing out of its head. Very strange. Now, in the 1970s, so that was after Watson, Crick, and Franklin discovered the structure of DNA, by the 1970s, we've got geneticists being able to actually decode DNA and write out the letters. DNA is only made of 4 letters, effectively, 4 different chemicals, which are just stacked together in different ways in your genome. And it was very laborious to start with. So they were writing out the genomes of different animals, and they thought they'd start with a fruit fly and write out the genome of the fruit fly. And it's all very well writing out a genome, but how the hell do you know what it means? You've just got this string of letters, A, C, D, C, 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 T, A, G, G, A. What does it mean? You don't know. And the way of working out what it did was to compare normal DNA with abnormal DNA. So here's the basis of this experiment then. You've got the wild type, the normal fruit fly genome, and you can lay that out. You can write all that out. And then you've got this strange mutant Antennapedia genome, and you can lay that out. And then you do a spot the difference. And where the DNA is different, that tells you where the normal patterning gene is, because that normal patterning gene has gone wrong in the mutant. So by studying the abnormalities, you uncover the normal pattern of the genes. And eventually, we end up with a whole series of patterning genes, which are basically telling, in the normal fly, there'll be a gene that's switched on in the head, which goes, right, you're the head, grow an antenna, effectively. In the mutant fly, something weird had happened to that patterning gene, so it went, I don't know, maybe you're part of the thorax, do you want to grow a leg? So this is a maggot, so this is a fruit fly embryo, which looks very different from our embryo, obviously. And this is a fruit fly embryo where that same technique has been used, where you can stick an antibody on anything you want and stick a coloured dye to it. And so we've got all these different coloured dyes, and you can see that those coloured dyes are clustering in the segments of this maggot. And what this represents is a different gene being turned on in each segment, or different sets of genes being turned on in each segment. So you can see at this one here, in this segment, you've got the product of this pink gene being switched on, and here's a green gene being switched on, and here's a blue gene being switched on. So what we're actually seeing is the way that these genes are influencing the patterning, the very early patterning of this developing embryo. So here's it in diagrammatic form. There's the chromosome, look, with the genes on it. And if this gene is being expressed, and what that means, it's being read in that particular segment, it's being read and a protein's being made from that gene, then you end up with that turning into the back of the abdomen. At the other end, if this red gene gets read and the protein from that gene is expressed, then that little segment is being told that it must become a head. And we've got a whole series of these patterning genes. They're called Hox genes. So this was elucidated in the 1970s. In the 1980s, geneticists moved on to going, well, are there similar patterning genes at work in vertebrates? So things which are a bit more like us, a bit more similar to us. And they looked at the mouse. And what they found in the mouse was 4 sets of patterning genes. They're all quite similar. They look as though they've maybe been duplicated over time. There are striking similarities. But they found 4 sets of patterning genes, each set on a different chromosome. And you can see a similar sort of thing. So, you know, if a segment's expressing this red gene, then it's going to be sort of probably around the neck of the mouse. If it's expressing these green genes, then that's kind of telling this area of the mouse to carry on turning into abdomen and tail. So it's interesting that we've got a similar sort of setup in this mouse. And if this mouse has got it, you've got it too. We're closely related. What's even more interesting is when you compare it with the fruit fly genome. Because the fruit fly genome is shown there at the top. So that strand at the top is the single set. Remember, fruit flies just have a single set of these genes. And the colours are really important here. Because the colours are not only showing you that, you know, these blue genes get expressed here, they're suggesting that there must be a similarity between these blue genes and that blue gene in the fruit fly embryo. There is. It's like reading the mouse hamlet is the blue gene in the mouse chromosomes, and the fruit fly hamlet, and it's the same play, maybe some of the character names have changed. And you're going, that's really odd, isn't it? Now, if you found 2 copies of Hamlet, which were very clearly the same play, with all the same intrigue happening, but with slightly different character names in one of them, what would you think? Would you think that somebody had independently come up with a complete, you know, this story of Hamlet, but with different names? Or would you think, I reckon there's an ancestor. I reckon there's a play that's been copied, and copied and copied and copied, and one copy's ended up with these character names, and one copy's ended up with those character names. That's what you'd think, wouldn't you? You wouldn't imagine that somebody came up with precisely the same play. And exactly, this is exactly what this information is telling us. It's not a coincidence that there's this striking similarity between the fruit fly genes and the mouse genes. The fruit fly genes and the mouse genes have come from the same place. They've been copied and copied and copied, and they've accumulated some differences, but they're still recognizably related to each other, and they're related through common ancestry. So a common ancestor 800 million years ago, it wasn't an insect, it wasn't a mouse, it wasn't any kind of vertebrate, it was some kind of worm that had these genes telling its segments what it would become in its embryo. And as I said before, with mice, if the mouse has got them, you've got them too. So you've got these genes, even though your body looks so different from a fruit fly, you've got essentially the same genes going head, neck, thorax, abdomen in your embryo. And then you end up looking completely different, but you've got the same program running. It's amazing. So the bones in your cervical spine were told to become that way. You know, they're all slightly different as we move down these segments by particular Hox genes. And then the thorax was told to grow slightly differently by different Hox genes. And then in the lumbar region, that area is being told to grow like that by different Hox genes. And then the sacrum, again, is being controlled by these Hox genes. And we've got layers and layers of these patterning genes operating. So that's just one set of genes. And there are many, many genes that are like this. So it's incredible because you can look at adult bodies and you can notice similarities between different animals and go, there must be an evolutionary relationship here. And it's very obvious with close relatives like chimpanzees. You look at chimpanzees and you go, okay, they look quite similar to us. They're hairy, okay, but their skeletons are very similar to ours indeed. The skeleton of the hand is impressively similar in a chimpanzee compared with the human. And that comes from a common ancestry going back about 7 million years ago. You can look at embryos, and that shows you surprising similarities because you haven't accumulated all the things which are special to you as a human. So in a human embryo, you will see those connections with fish, for example, which by the time you're an adult human, or even by the time you were a baby, you haven't got gill arches anymore, and those are only evident in the embryo. But then there's this deeper underlying vein of similarity that comes when we look at the DNA, when we look at the anatomy of the DNA. Let's just quickly see where this takes us. So we're week 5 with that human embryo, not a very pretty looking baby. Week 6, and arms and legs are growing a little bit more. The face is starting to form, but it looks very ugly to begin with. Week 7, we started to get a flattening out of the ends of the limbs, but in fact the toes and the fingers are still stuck together at the moment. By the time we get to week 8, this is where I think most of the interesting stuff's already happened. By week 8, you've essentially got a tiny baby. Strange proportions, but a tiny baby. It's got a beating heart. It's got a gut tube, and it's got a liver. It's got separated fingers and toes now as well, and it's recognisably human. An odd sort of human, but recognisably human. And then it's just got to spend another 7 months growing. And just going back to those pre-formationists, obviously their idea of a tiny baby curled up in the sperm or the egg is utter nonsense, but in a way they were slightly right. They were slightly right because their idea that the complexity was there from the beginning is true, but the complexity is not there in the form of the structure of a baby. It's there in the code for making one. So it's there in the genetic code right from the very beginning. We understand our development better through this science. We understand our relationship with other animals better as well. And we can start to say, well, we can create this tree of life on the planet by going, right, let's get us and our closest living relative, okay, chimps, so that's there, and then the next one's gorillas, and that goes back a bit further, and gradually you build a tree of life. In the 19th century, we had scientists trying to do it, and they did it something like this. And you'll notice that man is at the top. I think he meant humans, but anyway. Probably didn't, he probably just meant man. But, humans very much are where evolution is going. That's not our modern biological vision of evolution. Evolution just spreads out in lots of different directions. So when we draw trees of life today, in fact, they're more like Darwin's tree. So you've just got spreading out, lots of lineages spreading out. Nothing that's alive on the planet today is more evolved than anything else. We have the same depth of evolutionary history behind us. So you can represent a tree of life like this. It's still a bit parochial. We're still sort of featuring the animals with lots of cells in their bodies very large and downgrading the bacteria and the archaea, which actually are more diverse than all of the rest of us put together. This is probably one of the best ways to represent a tree of life because it's like King Arthur's Round Table. Nobody's sitting at the head of the table. Evolution isn't going in a particular direction. It's just pushing out all around the edges continually. So this is how we represent the diversity of life on Earth now. It does knock us off a pedestal, doesn't it? You're going, oh, I really thought that humans were what it was all about. And then you go, no, there you go. Homo sapiens. You're a twig. You're not the pinnacle. I love it, though. I love it, because once you've overcome that, moment of being humbled and going, oh, okay, so I'm not the be-all and end-all of the universe and life on this planet and all of that sort of thing. Look at where it does put us. We are so used to thinking about humans over here and the rest of nature over there. And this is modern biology saying to us, no, humans are in nature. We're part of it. We've evolved like every other species on the planet. And I think, you know, now that we've got to where we are with our capacities, our cognitive capacities, we understand the impact of what we do. In some ways, that makes it even more, it makes it imperative that we recognise our responsibilities and that we understand our impact on other species. And all those other species are our cousins. We've got to look after them better. But, yeah, we're leaving us here, a tiny twig on the beautiful branching tree of life on the planet. Thank you.